Welcome to this explainer on ProteoMind AI. Today, we're diving into an anti-gravity-based multi-agent workflow originally developed for the Kaggle and Google AI Agents-Intensive Vibe Coding Capstone project. We'll be exploring exactly how this platform automates the biological interpretation of proteomics-derived gene symbols. Let's get right into it.
Here is our agenda. We'll cover 1. The problem, 2. The ProteoMind AI solution, 3. The multi-agent workflow, 4. Outputs and applications, 5. Limitations, and finally, 6. The conclusion.
The problem.
So imagine you've just finished a statistical analysis and you have a list of significantly regulated proteins. Now what? Well, to actually understand their biological significance, researchers usually have to manually query a whole range of independent databases. You're going to UniProt for your annotations, Reactome for pathway enrichment, the Human Protein Atlas for spatial expression, and then over to String to build out interaction networks.
Doing this manually for dozens or even hundreds of proteins is incredibly labor-intensive. Constantly bouncing between different databases introduces a lot of repetition, increases the likelihood of human error, and frankly, it really hurts reproducibility. The goal here is to shift away from that tedious manual querying and move toward a streamlined, automated process, one that you can trigger with just a single Python execution.
Section 2. The ProteoMind AI solution.
It turns out that biological interpretation is actually a perfect use case for an agentic workflow. Instead of building one massive monolithic application, ProteoMind AI breaks the pipeline down into modular, highly specialized agents. Each agent handles a very specific scientific task, takes in structured input, and outputs standardized data. It's an approach that drastically improves both maintainability and automation.
Section 3. The multi-agent workflow. The workflow itself follows a logical, sequential journey. First, you input your curated list of human gene symbols. From there, the data passes through 3 specialized agents. Agent 1 handles the UniProt queries. Agent 2 takes care of Reactome and the Human Protein Atlas. And finally, Agent 3 steps in to communicate with String.
What's really efficient here is that by integrating existing database skills within the anti-gravity ecosystem, the system avoids the need for custom database clients. In this structure, Agent 1 annotates metadata, like GEO terms, using UniProt. Agent 2 follows up by pulling pathways from Reactome and spatial localization data from the Human Protein Atlas. Finally, Agent 3 constructs those high-confidence protein-protein interaction networks using the String database.
Section 4. Outputs and applications. The final outputs are cleanly formatted and immediately ready for downstream interpretation. The pipeline automatically generates neatly organized CSV files for your annotations, enrichment scores, protein-protein networks, and significant Reactome pathways. But on top of that, it also compiles markdown summaries for pathway enrichment and interactions, giving researchers a clean, ready-to-read overview right out of the box.
Now, even though this was originally built with differential proteomics in mind, its applications are actually quite broad. Because the workflow accepts any curated list of human gene symbols, it's just as effective for RNA-seq data, biomarker interpretation, and broader functional genomics studies. And a nice bonus? It doesn't even require both up- and down-regulated proteins to function properly.
Section 5. Limitations. Of course, it is important to address the constraints of the current workflow objectively. ProteoMind AI is exclusively focused on downstream interpretation. It won't perform differential expression analysis for you, nor will it calculate fold changes or p-values. Furthermore, it strictly requires mapped human gene symbols, is validated exclusively for Homo sapiens, and naturally, its biological accuracy depends entirely on the confidence scores of those external databases.
Section 6. Conclusion. Ultimately, the big takeaway is that rather than trying to replace established, highly trusted biological databases, ProteoMind AI seamlessly orchestrates them. It turns them into a single, reproducible workflow using specialized AI agents, simplifying and accelerating discovery while maintaining strict scientific rigor. And because of that modular design, this architecture is practically built for extension. So looking ahead for ongoing GitHub and Kaggle development, we have to ask, what new identifier mapping utilities or organism-specific workflows could be seamlessly integrated next? The foundation is certainly there for a highly adaptable future in automated biological research. Thank you so much for joining me for this explainer.